Hi, Farasi, Falguni and myself will be presenting how insulin binds. Binding of insulin to the insulin receptor plays a central role in the hormonal control of metabolism. Here the authors of the article, How Insulin Binds, investigate possible contact sites between the receptor and the conserved non-polar surface of the B chain. Evidence is presented that two contagious sites in an alpha helix, valine B12 and tyrosine B16, contact the receptor. Substitution of tyrosine B16 by an isosteric photoactivity derivative, also known as para-azidophenylalanine, which can be referred as PAP, enables efficient cross-linking to the receptor. Together, the results support a model in which the B chain alpha helix functions as an essential recognition element. Insulin is a globular protein containing two chains, A with 21 residues and B with 30 residues. Stored in the pancreatic beta cells as zinc ion stabilized hexamer, the hormone binds to its receptor as a zinc ion free monomer. Critical side chains are located both in the hydrophobic core and at the protein surface. The present study focuses on two conserved sites on the protein surface, valine B12 and tyrosine B16, which is shown in green in figure 1A and B. Figure A shows the ribbon model where A and B chains are shown in dark and light gray, while figure B is a surface representation of human insulin where the sulfide bridge is shown in yellow. The figure C shows a schematic structure of insulin receptor, whereas figure D shows the chemical structure of PAP, however, only the side chain is shown. Insulin binds to and activates a receptor, tyrosine kinase, known as TK, on the surface of target cells. The insulin receptor is a disulfide tetrahedral alpha-2 beta-2 heterotetramer, while the beta subunits are transmembrane proteins containing intracellular TK domains. We shall first see the presence of a tyrosine on the surface of central B chain alpha helix tyrosine B16 to introduce the aromatic derivative PAP. We see that PAP B16 undergoes rapid and efficient photo-cross-linking to the insulin receptor. Later on, we shall investigate the structure-function relationships at position B12, a neighboring site on the surface of the alpha helix. The results strongly support the hypothesis that valine B12, like tyrosine B16, directly contacts the insulin receptor. Falguni will now talk about the results.
The results are split up into two parts. Part 1 is about the studies of insulin containing a photoactivable amino acid derivative in the central alpha helix of the beta chain. Part 2 is about defining the structure-activity relationships at positions B12. However, mainly we'll be focusing on Part 1. During this experiment, two derivatives of DKP insulin containing PAP substitutions at position tyrosine B16 and phenyl B25 were prepared by chemical synthesis. Figure 4A and B show the analysis of chemotypic digestion of PAP B16 and PAP B25 DKP insulin cross-link complexes, which is showed using a Western blot. Aliquots were then stopped at the indicated times. Control studies of the PAP B25 insulin analog were then extended to studies of a pre-trypsinized but active receptor. Digestion of the cell-bound receptor yields a collection of cleaved receptors, including an active derivative containing a C-terminal cleavage from the C-terminus of the alpha subunit, which is shown by the black arrow in figure 4C. At lanes 5 and 6 in figure 4D, there is a cross-linking of PAP B25 DKP insulin to the pre-trypsinized receptions, which produces two bands, the intact alpha subunit and an additional band near 37 kilodalton. The red box in lane 6 indicates absence of such cross-linking in the PAP B25 complex. The asterisk shown in red in lane 4 indicates cross-linking of the B16 analog to a fragment of alpha subunit lacking the C-terminal 28 kilodalton site. Figure 5 shows the electrophoresis represented gels using a Western blot used to map sites of cross-linking in the alpha subunit of the insulin receptor on limited tryptic digestion. It further defines the B16 binding region, which shows the limited digestion of the covalent hormone receptor complex. A unique amino terminal 31 kilodalton cross-linked glycosylated fragment was hence identified in PAP B16 cross-link complexes in lanes 1 to 4 in figure 5A and B, but not in PAP B25 cross-link complexes in lanes 5 to 8. Figure 5D is a homology model of the L1CR-L2 sites, which predicts exposed tryptic sites at positions K149, K164, and K166. Grazi will further talk about the discussion.
Classical studies of structure-activity relationships suggest that the crystallographic dimer interface defines a major part of the monomer's receptor binding surface. Although hexamers and dimers do not themselves bind to the insulin receptor, interfacial residues define an extensive nonpolar surface in an insulin monomer. Substitution of tyrosine B16 by a molecule with a side chain of similar size, like PAP, was found to enable efficient studies. The insulin and insulin growth factor bind to a receptor with opposite orders of affinity. Chimeric receptors based on mixing and matching domains have enabled determinants of ligand specificity to be identified. Such studies suggest that L1 domain allows high-affinity insulin binding. The present study demonstrates a specific role in docking of the central B chain alpha helix. After the discussion, we concluded that how insulin binds to the insulin receptor represents a major unsolved problem in structural biology. The present study has demonstrated that the central alpha helix of the B chain contacts the L1 beta helix of the insulin receptor. High affinity and low affinity contacts are defined at residues B12 and B16, conserved sites on the surface of this helix. Valine B12 and tyrosine packet the nonpolar dimerization interface of the free hormone and are likewise proposed to bind at the nonpolar surface of the receptor L1 domain. The approach demonstrated here is likely to generalize to define additional sites of contact between the hormone and its receptor. Further, a structural understanding of insulin binding and signaling would make possible a design of a new supporter for the treatment of diabetes mellitus. Thank you for listening.